Hello, everyone, and thank you for joining us today. My name is J.C. Hernandez, and I'm a tech transfer analyst at NASA Johnson Space Center. I will be your host for this NASA Technology Transfer Program webinar on the technology self-contained device isolates biological samples. Our presenters today are Dr. Honglu Wu and Dr. Yi Tseng. Dr. Honglu Wu is the manager of the Radiation Biophysics Laboratory within the Human Adaptation and Countermeasure Division at the Johnson Space Center. His research activities include investigations of the combined effects of space radiation and microgravity in cells and animals. He is also interested in technology development. The JSC Center Technology grants that Dr. Wu received include carbon nanoparticles as biomedical countermeasure against space radiation exposure, and an organ-on-chip device for investigation of drug metabolism in the microgravity environment, among others. Dr. Yi Tseng joined the Radiation Biophysics Laboratory at NASA Johnson Space Center in 2007. After postdoctoral training at Baylor College of Medicine and UT Health Science Center in Houston, Dr. Tseng became the lead senior scientist of the Bioanalytical Core Laboratories at JSC in 2009. Her current responsibility is to support the NASA Space Biology Program within the Biological and Physical Sciences Division under SMD to develop plans and strategies to engage space biology researchers and to manage space life sciences missions and grants. Following the presentation on the technologies, I'll be giving a short presentation on how NASA licenses technology to outside organizations. Before we get started, I'd like to point out that your microphones will be muted throughout this presentation. So if you have any questions, please type them into the chat box in the lower right corner of your screen, and we'll answer them during the Q&A session at the end. At this point, I'm going to turn it over to you, Dr. Wu and Dr. Tseng. Dr. Tseng, if you would just let me know when you'd like me to forward the slides, then I will happily oblige.
Right. Good afternoon. So thank you, JC, for giving us this opportunity. Yes, can you just please forward to the next slide? Yeah, so this is just introduction. I think thank you for JC to introduce me and Dr. Wu. And then, yes, thank you. So just here's a background. I think everybody in this webinar definitely know that the ISS, the International Space Station, has been designated as a national laboratory dedicated for biomedical and biological research for the benefit of human health on Earth. It is essential to establish the capability, the state-of-the-art capabilities to analyze biological samples like DNA, RNA, proteins, and cells in space to understand how cells respond to the space environment. So microbial contamination, that is another concern, and infection pose a significant risk to the health of the astronaut and to the health of the hardware and structural materials on the ISS and during future missions to the Moon and Mars. So a capability to identify microorganisms in space allows effective and immediate treatment in order to ensure the crew health and the success of the mission. Yeah, next slide, please. So you can see this, this picture that actually are from the ISS sample. So this is quite an old statement in a way, but it's still the same. The primary justification for support of the ISS is its scientific and research potential. And also the same for the future commercial, space station. And then the NIH ISS program now is like cases, the National Lab NIH program focus focuses on the biomedical research on the molecular and cellular level that will use the unique microgravity and radiation environment on the ISS and resources available on the ISS to test the innovative hypotheses for the potential benefit of human health on Earth. Next slide. Then before we go to the technology development, why we focus on the DNA RNA isolation and the PCR technology, enabling PCR technology on the ISS. So I want to introduce a little bit about PCR, real-time PCR, polymerase chain reaction. The real-time PCR is not a brand new technology, as has been there for years. However, PCR, real-time PCR or PCR actually is a very fundamental technology that has been used and will be used for all the different analysis, molecular analysis, subsequent downstream analysis, like even sequencing, single cell sequencing, the microRNA sequencing, RNA sequencing, DNA sequencing, mutation detections, and you name it. So all those states of the art molecular omics technologies actually based on the PCR. We need to prepare the sample using the PCR technology. Then we can go from there to sequence the sample or to conduct the downstream analysis. So real-time PCR is one of the most sensitive and widely used instrumentations or technologies for modern molecular biology analysis, including quantifications of DNA, RNA targets. So for diagnostic applications, real-time PCR is well known, can be applied to rapid detect nucleic acids that are associated with infectious diseases and genetic mutations. Well known right now, we have COVID. So actually the PCR method to detect a COVID infection was actually the first line development, I think in 2020. So it was a PCR technology was significant, has significantly improved the diagnosis of infectious disease and is deployed as a tool to detect newly emerged diseases. So in research settings, real-time PCR is widely used to provide quantitative measurements of gene transcripts in response of tissues and cells to changes in environmental conditions, such as those conditions on the ISS, altered gravity and radiation environment. So the right side, you can see that, just a little bit about the PCR technology. So we have, for example, we have a DNA double strand. We have a DNA molecule that has a double strand. And then when you do the PCR, the first step will hit the double strand DNA, and then to separate them and generate single strand DNA. And then it has primers, specific primers to the gene target, and then the primers will bind to the single strand DNA, and then you need to add the polymerase, and then the DNA polymerase will extend the primer to completing the whole sequence, the new DNA molecule or DNA strand, and then to generate 2 DNA molecules. And then after that, we'll have another hit cycle, and then to separate them to start the next cycle. So each one is a complete cycle. So for example, if you have one DNA molecule target at the beginning, then after 30 cycles, you can imagine how many DNA molecules for the same target and then can be analyzed. You can be able to read the signal. So that's a very sensitive and powerful technology. Next slide, please. So applications of real-time PCR in space can be, widely used. It can be applied for many type of analyses. So, for example, to detect the infectious pathogens and the environmental contaminants, to monitor the time course drug-resistant microbial and dangerous mutations, those development, and then to identify new genotypes and types of microbial and possibly new species, and then to investigate the effects of space flight factors on living organisms at the molecular level. So next slide, please. So that we can apply those PCR actually not only on the Earth, on the ISS, also on the lunar base, and even further when we go to Mars. So technical, we do have quite some technical opticals in performing real-time PCR in space. Actually, the major one is a sample preparation. So for a PCR reaction, we need about 5 to 6 different reagents, different type of the solutions. In addition to that, we need to have we need primers, we need enzymes. They kind of a different storage condition, under different storage condition. And also we need some, the way to introduce a sample. And we also need certain binding membrane to bind specifically to the molecules. So the sample preparation is a major optical. Because on the ISS or in the space for future missions, we don't have hazard-free and microgravity compatible hardware for DNA RNA isolation, especially automated one, and for the PCR preparation. So as I mentioned earlier, actually, in addition to what I mentioned, 5 to 6 reagents and different types of the enzymes and different steps. Each step also requires multiple pipetting, reagent mixing, centrifugation, disposal of hazardous reagents. And modification of the real-time PCR machine is needed to ensure proper operation in the microgravity environment. Actually, this one has been, we had already made quite some progress on the PCR machine side. So can you move to the next slide, please, JC? So the goal for this project, actually the whole project was actually dated back to about 2011 to 2013. So the goal at that time was to develop a DNA RNA isolation and a PCR preparation kit to be self-enclosed and pipette-free for use in space and on Earth. And sample isolation and preparation will be accomplished, or kind of like the concept is to use a set of connected syringes preloaded with appropriate solutions and binding membranes that capture the desired molecules such as DNA, RNA, or even proteins. Next slide, please. So here is show our prototype. Left side is a drawing that's or figure 1 actually in the pattern. And then when we think about how to put everything together, 5 to 6 reagents, different solutions, and binding membranes, the method to introduce the samples, and how we can connect, because how we can moving those solutions around, and then in an enclosed device, and then to deliver the final product. So when we look at all the possibilities, all the options, actually we found that if we put 2 multi-way valves, actually this is a 4-way valves that you can move all different directions. So if you couple those two, and in between you have a compartment, and then you coupled with the two four-way valves, so that gave you maximum or at least a significant increase of flexibility. How you can moving around the liquid, the reagents for different directions, how you can be able to isolate the sample or concentrate the sample at a certain membrane, a certain compartment, how you can put the waste into some waste container. So that gave you a really good flexibility, how you can moving those reagents around. So the right side is the our prototype. Actually put all the parts together and then to make it real. And then next slide, please. It looks very simple. And then, so we did some validation test. And then right side, the middle panel, you can see that it was a comparison of efficiencies of DNA isolation using the prototype. And then they actually compared to the traditional way, like using centrifuge, pipetting, and all these traditional ways. And they actually, they generate a similar amount of DNA when we use the same cell number using two different methods. And then the upper panel, actually, we used the plasmid DNA to look at whether we can recover. We add plasmid DNA and then see whether using this device we can recover the DNA. So actually, we got quite comparable, quite even better results than the traditional method. And then if you look at the lower panel, and that we call elution, that means we bind the DNA to the membrane. And then normally, when we elute DNA to the solution, we have to centrifuge. We have to add the solution to the membrane and with a tube and centrifuge, and then to elute the DNA. But using this device, actually, we really do not need to elute, and we got the result comparable to the traditional ways. Next slide, please. So the phase 2 for this development is really, the effort to develop automated devices. We put here, we developed a kind of semi-automated devices. We have not done the validation test for the DNA isolation, but to be validated how the fluid and the connection and the movement and everything that is as what we designed, expected this unit to be able to. So the outcome is actually we developed this semi-automated device that potentially enables preparation, isolation, and analysis of biological samples for microgravity or some other environment, like some extreme, hostile environment, or even for military use. So this summary is this self-contained device for isolation DNA, RNA, protein, and cells is a component system that includes syringes, pistons, membranes of different capacities, reagents, multiple valves, and small pumps. By combining the valves, the multiple valves, you'll be able to moving things around. Actually, gave you really maximum flexibility, expand the capability of the capability of how we can prepare the sample for space usage, space application. So, okay, go to next slide, please. Yeah, we still keep to, our intention is to still use the conventional, the reagents. So we don't want to change the reagents because those are gold standard, like, has been tested, validated, widely accepted, proven to be a high quality, we still want to use those methods. We just want to have the self-enclosed device, more automated device. We can put those reagents together and then deliver the good quality samples for the for PCR and for downstream more complicated omics or other type of applications analysis. So this is some possible future development. So the A actually represents the current functional unit of a sample preparation system rearranged from the semi-automated platform. The syringes are connected to four-way valves. And then B is the multi-stacked sample preparation system with syringe plungers and the valves controlled by motors. And then the C is the system would be controlled by the software developed to process different samples. Basically, can be programmed for different type of sample preparations. And then D is the a kind of a commercial product. That's a future commercial product. That's our vision to be used in space, but also can be used in other areas, environments or applications. Can I go to the next slide, please? Thank you. So other applications. So this, even though we mentioned a lot about the DNA, RNA isolation, the PCR, actually this can also be used, have to modify it, but can be used for other molecular analysis, such as using sequencing technology or instrumentations like a microplate reader. Because no matter what kind of technology, what kind of instrument, what kind of the analysis, they all need sample preparation. So that's a step we cannot skip. And then also that's a step it's not easy, really not easy to be achieved in space. Can I go to next slide, please? Thank you. So this is the other one, that this device can be modified and used to prepare samples for in-orbit and orbit and other kind of analysis with instruments like a flow cytometer, for example, by performing immunofluorescence staining and separating different cell types. So this is our latest semi automated system. And then you can see that at the front there are two valves, micro valves, multi-way valves. And then in between, actually, it's a compartment that we can use that for the like a binding membrane. But if we put like a cells chamber there, actually can easily fix the samples, keep the culture going, fix the samples, stand with the samples, and then prepare the sample for the, for example, flow cytometer analysis. So I would say this whole concept is kind of a flexible. The concept can be used for many different applications. So go next slide, please. And then this, as I mentioned earlier, and this device can be modified to perform experimental procedures on Earth and in orbit, such as doing cell culture, medium change treatment, fixation of cells in order to reduce the crew time and improve the quality of the experiment, consistency. So this is one of our previous ISS experiments. And the hardware actually still, this hardware still is used now by the researchers. And if you look at that, in the middle, that is a tissue culture chamber. and then that use a two-way valve, and that only can have a waste bag, a PBS bag, and then maximum two reagents. I remember one we used for induce a treatment and then inject a treatment solution, and the other one was used for the fixation. That's a maximum for this. However, if you use a modified OR device to make this, can maximum what we can do over there, especially for more complicated biomedical research and many other applications, more advanced tech, more advanced research. We need really enhance. We really need to enhance our capability right now on the ISS to be able to accommodate those type of research. Yeah, next slide, please. So here's the technical highlights, just mainly for the DNA RNA isolation part. So this so far our technology or development, we can using our development, we can isolate DNA RNA without using pipettes or centrifuges. And then this device is self-contained and includes reagents from the membranes with different type of capacities and the multi-way valves and pumps. And due to self-enclosure, devices leak-proof, so users are protected from hazardous reagents. And also I want to mention, because of that's a very small volume, like hundreds microliter, if used for RNA, DNA isolation or protein work, so the waste actually also microliter level. It's very minimum, the waste volume. And then this system, this device also is developed to potentially enable full automation and the software programming, extending laboratory isolation protocols to applications especially for space usage. And then the development itself actually the concept actually is a device is microgravity compatible, but we have not tested yet. I think at least a parabolic flight test could really tell us a lot more, validate whether it is microgravity compatible. And then the potential, also this technology is potentially used for field biology or other remote clinical operations, military setting, military usage. And then so for future development, multiple kits can be stacked to allow multiple samples to be processed simultaneously, and the system is very flexible and can be modified and used in for other existing analysis modules, instruments, and other applications. Next slide. I think that's the end of the presentation.